last time we did the basic structure of the cow and how basically the gastrointestinal tract works. This time we're going to talk about carbohydrates. Okay, and when we talk about carbohydrates, there's two types of carbohydrates we worry about. Normally we worry about structural and non-structural. Okay, and so your structural carbohydrates are basically what provides plants with structure. So when we think about that, we would think about like grass, legumes, hay, silage would contain a lot depending on the type of silage. So anything that holds the plant up, it's made primarily of cellulose and hemicellulose. Okay. So for structural carbohydrates, we're going to normally use green because green makes me think of grass. And for non-structural carbohydrates, we're going to use red just because it will show up. Okay, and when we think about non-structural carbohydrates, what we're talking about there is grains, namely. So we can think of corn, milo, etc. So anything that contains a lot of sugars and starches. So other feeds that might be a non-structural carbohydrate or a good source of that would be molasses or any kind of liquid feed would tend to contain a lot of sugars and starches. And so how the animal is going to use these are different, but we'll start with the similarities. So again, we have the mouth, we have the rumen, we have the small intestine, we have the large intestine, we have the pancreas, the gallbladder, and the liver. Okay, so if our cow is grazing and she consumes some structural carbohydrate, it enters the reticulum rumen, and one thing about structural carbohydrate is she is going to ruminate it, so it goes both ways multiple times. So one thing to keep in mind with structural carbohydrate is structural carbohydrate increases rumination. Okay, which if you take a minute and think about it, takes a whole lot more time to break grass into little pieces. If I gave you a pile of grass and asked you to break it apart or rip it apart, it would take a lot more time than if I asked you to take corn and break it into little pieces. Okay, so we feed the animal structural carbohydrate. The animal could also consume non-structural carbohydrate, and it will also enter the rumen. Okay, and it is also ruminated, but not to the same extent. So we're going to make that arrow a little smaller because less rumination occurs with non-structural carbohydrate than with structural carbohydrate. Okay, there are different types of microbes. Last time we talked about bacteria, protozoa, and fungi. Okay, and all three of them work on both structural and non-structural carbohydrates to some extent. But we have different populations that work on structural carbohydrates. So you have cellulolytic microbes, and those work on your structural carbohydrate or those that contain cellulose. And you have your amylolytic microbes, and they work primarily on starch. Amylose is another name for starch, so they work primarily on starch. So what we end up with is we have fermentation occur for both of them. And that produces VFAs. and those VFAs are absorbed out of the rumen. Okay, the other thing we end up with is microbial crude protein plus CO2 plus methane. Okay, the CO2 and the methane are eructated, okay, they're burped out of the animal. And as we said before, methane and CO2 both, but methane in particular is a greenhouse gas that there's a lot of concern about. And with structural carbohydrate fermentation, we tend to get more acetate, which favors methane production. Okay, structural carbohydrate in general also tends to be less complete because digestibility is lower. Okay, and so in general, structural carbohydrates, we have increased rumination, more acetate, and less complete digestion because digestibility is lower. Reason we feed structural carbohydrates is they tend to be cheaper, and cattle tend to be the most efficient utilizers of those structural carbohydrates of the animals that we use to produce meat and milk in the United States. Okay, if we look at non-structural carbohydrate, we tend to have a decrease in rumination, an increase in propionate. Okay, and that's a positive thing, right? Because we said last time that propionate provides glucose and that can increase the marbling in the animal. So this is one of the reasons why we feed non-structural carbohydrates to cattle during the finishing phase, is because they're going to use that to produce more propionate, which will increase marbling. So increases marbling and decreases methane production. And the reason it's going to decrease, that's a 4. This is a 4. It's going to decrease methane production is propionate's a hydrogen sink, which means it accepts hydrogen during fermentation. And so that steals hydrogen away from methane. So a real positive is a decrease in methane. And they tend to be more digestible.
Can't see that.
They tend to be more digestible because of just the way they're built and the way they're structured. They're not associated with lignin. So one thing we need to add. Yeah.
Move it down a little bit.
That's good.
Is it okay?
Mhm.
See everything again? Sorry.
That's okay. Okay. An additional thing about structural carbohydrate is they tend to be associated with lignin. Okay, and lignin is not a carbohydrate, it's a phenolic polymer. It is not a carbohydrate, but it tends to be associated with hemicellulose and cellulose, and the presence of lignin really decreases digestibility. And so lignin, you'd be most familiar with being present in wood, and there's nothing that really digests lignin in the cow's rumen. So lignins are real negative from digestibility. When we look at non-structural carbohydrate, there's no lignin associated with it. The other thing that makes non-structural carbohydrate different is it tends to be in seeds or cell contents, and the plant actually had plans to utilize this carbohydrate as a source of energy. So corn stores starch in the corn seed, so it has energy to grow corn. Okay, if there is some structural carbohydrate that doesn't get fermented in the rumen, it goes through the small intestine basically untouched. Okay, and in the rumen we can ferment it again and produce VFAs, and those VFAs get absorbed and go to the liver. Okay? And we also get the production of MCP, CO2, and CH4, all of which get excreted in the feces. Non-structural carbohydrates are a little different in that the pancreas produces enzymes, and the main enzyme that we're worried about the pancreas producing this time is pancreatic alpha amylase. Okay, so what we have occurring is we have the pancreas producing pancreatic alpha amylase, and that's released in the small intestine. The small intestine itself also produces some enzymes. We'll just call these starch digesting enzymes. We're not real interested in who they actually are and what they actually do. But the end result is we have the release of glucose. Okay, and that glucose is absorbed by the small intestine. Okay, and really the only non-structural carbohydrate we're going to expect to see is starch. Okay, if we had a baby ruminant or a baby calf or a baby lamb, we would see some milk, but that's a different thing and kind of a different process. So some starch will not get digested in the small intestine and will be used to produce microbial crude protein, CO2, methane, and more VFAs. And once again, those VFAs favor propionate, right? Because we have more propionate. So when we look at the digestion of carbohydrates, we have really only two things absorbed. We have VFAs absorbed. We have glucose absorbed. So VFAs are absorbed from the rumen, and VFAs are absorbed from the large intestine, and then glucose is absorbed from the small intestine. We have microbes produced in the rumen, microbes produced in the large intestine, CO2 in the rumen, CO2 in the large intestine, CH4 in the rumen, and CH4 in the large intestine. Microbial crude protein, CO2 and methane get excreted in the feces in the large intestine. CO2 and CH4 get eructated out by the cow as gaseous energy loss. Okay, the VFAs go to the liver, and so we'll draw that real quick, even though we drew it last time. Okay, and what the liver is going to release is acetate, ketone bodies, and glucose. Okay, and that's all from the VFAs. This glucose down here is also goes to the liver. My red marker is dying. And we're going to release glucose from there as well. Okay, and then these three things, acetate, ketone bodies, and glucose, are going to be used by the body to meet the energy requirements of the animal. Okay, so whether it's structural carbohydrates or non-structural carbohydrates, you end up with acetate, ketone bodies, and glucose. Non-structural carbohydrates favor propionate, and we also get glucose from the small intestine. So non-structural carbohydrates are going to provide you more glucose because they provide propionate, and also because some glucose is going to be absorbed out of the small intestine. But most of it is still going to be fermented in the large intestine. Just a small amount gets absorbed as glucose. Okay, so basically what's going to happen is, or what we've talked about today, is how the microbes utilize structural and non-structural carbohydrates. And what we're going to talk about here in a few minutes is how protein is involved in that and how microbes require protein to be able to ferment non-structural and structural carbohydrates into VFAs. Okay?